Good afternoon, everyone. So once again, I welcome you all to this practical session. So if you go by your manual, so our first experiment is expression analysis of a gene, right? So we are going to analyze the expression of a gene using a very potent method that is called as quantitative real-time PCR. Quantitative real-time PCR. So if you recall some 5 years back, so northern blotting was one of the most fab method to be used for the expression analysis. But because of use of radioactivity and then a lot of tedious process, so it was somehow shut out, and now we are in an era where people they use real-time PCR for the expression analysis of any gene. So this real-time PCR, actually there are different synonymous terms which are used for this quantitative real-time PCR. For example, we also used to call it as kinetic chain PCR. We also used to call it as QRT PCR. We also used to call it as RTQ PCR. And sometimes it is also notated as capital Q. So these are some of the notations which generally if you go by the publication, so people they are using different notations to designate the quantitative real-time PCR. So now first and foremost thing which we have to understand is what is the utility or use of this particular technique. Until unless you know the use, you cannot decide where to fit this. So this particular quantitative real-time PCR is used for, first is as I told, expression analysis of a gene. This technique is also very popularly used for the validation of transgenics. So this is one of the accepted method other than the southern blotting method for the validation of transgenic. And in foreign countries and in good labs, people they are using real-time PCR for the estimation analysis of nucleic acid also. Like here you are using nanodrop or you are using qubit, but in foreign countries this real-time is very commonly, routinely used for the estimation of the concentration of nucleic acid. Other than this, nowadays this real-time people now they have started this for characterizing the pathway by analyzing the expression of a gene, by identifying some of the mutation in the sequence. So that also you can easily analyze using the real-time PCR. So these are some of the important uses where you can fit this particular technique. Now, next question is, if I wish to use this technique, what are the requirements? What are the things which you require so that you can assemble all those and then you can go for real-time PCR? So requirements for this particular. First and foremost thing is the platform, the machine which you are going to use. This is called as platform. So in case of real-time PCR, basically we have two popular types of platform. One is called as closed platform. Other is called as open platform. So in case of closed platform, which was like maybe 5 years or 10 years back, all the real-time PCR machine was closed system only. So closed platform means whatever accessories you are going to use, that you have to purchase from the same company itself. There is no liberty that you can purchase from some others because that will not fit into your platform. So that is called as closed system. Open system means there is no restriction. You can have the accessories from any companies of your choice. There is other difference. So closed system, in case of closed, halogen bulb was used as a detector. So if you are using halogen as a detector, there is a limited hours fixed with all these halogen bulbs. Like you can use it for 1000 hours or 1200 hours. But in case of open system, we have a LED based detectors. So LED based means minimum 25 years. Up to 25 years you can use, and after that you can replace this. But there is a disadvantage also. When you purchase halogen based detector, that will cost you around maybe 60,000 rupees. But when you go for LED based detector, that will cost you around 4 to 5 lakhs rupees. Other disadvantages, in case of LED based detector, even if one LED goes out, you have to replace whole. But in case of halogen, if it is not working, easily you can replace it with another halogen bulb. Okay? And one advantage is open platform system has a very high equipped software, so they will analyze and they will give you result in a usable form. But in case of closed system, so you have to take the result out and then you have to do characterization on your own. Right? So first thing is platform. You should have access to open or the closed system platform. Second is high quality total RNA. Right? Because we are talking about expression of a gene. So here template you are going to use cDNA, and for cDNA you need to have a high quality of RNA. Now the question which comes in our mind is how to get high quality total RNA. Right? So there are different kits, but the most robust method which has been preferred for total RNA to be used for real-time PCR is trizol method. Other than trizol, if you are not equipped or if you don't have access to trizol, so you can use any of the kit. A lot of kits are available from reputed company. You can use those kits to isolate high quality total RNA. Now I am using the word high quality. So the next question is, sir, how to analyze the quality of this RNA? How we are going to analyze? So there are different methods to analyze the quality of the total RNA which you have isolated. The first and best method is gel-based visual method. The first is gel-based method. And other than this, we have series of methods like you can use qubit. Qubit is a fluorometer which will give you the quality in terms of ratios, right? Then you have a nanodrop. You can use nanodrop. This will also give you the concentration, and other than concentration, it will also give you the quality of the particular nucleic acid which you have isolated. Other than this, if you go higher end, you can use bioanalyzer, right? So there are different companies, they have their own bioanalyzer. This will also give you the concentration as well as the quality of the nucleic acid. And now we have a diamond moth-based capillary system, right? It is just based on the emulsion. Just use a drop of your nucleic acid and it will give you all the history of that nucleic acid in terms of quality as well as concentration, and the accuracy is far better than that of all other instruments which I have listed here, right? This is capillary based, so there is a small capillary tube and there you are going to put one drop of your nucleic acid and it will travel, and based on that, so they are going to characterize, right? So once you analyze the quality of the RNA, next is cDNA synthesis. The most critical part in case of real-time PCR is cDNA only, because if your template is not good, so you cannot expect a correct result. So this is most critical part. So cDNA should not be truncated. It should be not a mix of small and large fragment. It should be homogeneous, uniform, and it should be of a large quality cDNA. So here you have to check, you have to use a kit which is a reputed one and then which has more efficiency. Some of the kits like Fermentas kit is very popular. Even Takara, they have their own cDNA synthesis kit, which is very good, and a lot of labs they prefer, right? So again here, if you want to quantify, if you want to analyze the quality, so first is gel based. So when you run this on the gel, So we expect that you should get a smearing, and other than this, prominent band should be there. If it is a thin smearing with a prominent band, it means your cDNA is of high quality. If it is complete smearing, it means you have degraded out, right? And then when you analyze it on the instrument, so based on the ratio, easily you can find out whether your cDNA is good or not. Is it okay? So once cDNA you have synthesized, next is the chemistry which you are going to prefer. So in case of real time, two very popular chemistry are there. One is cyber-based expression analysis. We also used to call it as. So cyber is basically a cyanine-based dye. It is a water-soluble dye. So cyber-based chemistry, which is very commonly used for the expression analysis of a gene. Other is Tachman-based chemistry. Tachman-based chemistry. So I'll just brief you about differences between these two chemistry, cyber and Tachman. So the first difference is that in case of cyber-based chemistry, we are going to use a pair of primer, that is forward and reverse primer, which is gene-specific primer. Here we are going to use a pair of primer, that is forward and reverse, which is basically a gene-specific primer. But in case of Tachman, other than the forward and reverse primer, we are also going to use a sequence, right? We are also going to use a specific sequence which hybridizes with the gene. We are going to use a specific sequence which hybridizes with the gene, and it has a specific endpoint. It has a specific endpoint like this, with a fluorophore and a quencher. With a fluorophore and a quencher, right? In case of cyber-based chemistry, the expression which you are going to analyze is very relative one. It is not so accurate. It is just approx expression, so it will not give you the exact expression of the gene. But in case of Tachman, since this is per template basis, so whatever expression you are going to get, that is exact one. Accuracy is very high. Accuracy is very high in case of Tachman-based chemistry. Okay. Cyber-based chemistry, the cost per reaction is very low. Means it is user-friendly and cheap. Tachman cost is very high as compared to cyber. Tachman cost per reaction is very high as compared to cyber, right? Cyber-based chemistry, so it doesn't require much expertise. If anybody knows PCR, easily he or she can keep the reaction. In case of Tachman, you require the expertise to design the sequence, then only you can, it required expertise to design the sequence, and then only you can execute, and it required a proper training. Then only you can go for Tachman-based chemistry, right? And last is, in case of developing countries like India, very few labs are there who prefer Tachman-based chemistry because of its tedious way and then cost. But cyber, in almost all the labs, people they are using cyber for the expression analysis, right? Some of the key players of this Tachman and cyber are like Invitrogen, which is proprietary of Thermo Fisher Scientific. So this is very good company, and they have expertise in designing this. Then you have Sigma, so they have also expertise, and they have their own product of cyber, which is very cheap as compared to others. So that you can prefer, you can purchase, and you can use, right? So once you decide which chemistry you are going to use, then the most critical point comes, that is the oligos. So when you go for the oligo designing, there are few criteria which you have to follow, then only you can design the oligo. So some of the specific features which you have to follow, or you have to put into the software for a good primer to be used for the expression analysis are like length. The length of the oligo which you are going to use for the expression analysis, optimum it should be 18 to 20. It should not be very lengthy, 22, 24, something like that. 18 to 20 is the optimum length of the oligos. Then when you talk of the TM, most of the time we prefer that the TM which you are going to select should be 60. Then your primer should not self-anneal. It should not end with a base more than twice. Means at the starting or the end point, there should not be same base more than 2. It should not be 3 or something like that. There should not be a complementarity between the forward and the reverse primer. It should not form a dimer. The primer which you are going to use for the real-time expressions should be HPLC purified, right? Many times common primer we are using and then expression will be very bizarre type. You will not able to correlate. It should be HPLC purified. So let me repeat once. I told length 18 to 20 that you can prefer. TM it should be 60. A few people they prefer to have a 58 as a TM, but then for cloning 58 is the optimum. For expression analysis people prefer to use 60 degrees Celsius. Then all others that I have told. So when you take all these precautions, so some of the softwares which are freely available where you can design primer for real-time PCR expression analysis are like you can use Gene Fisher primer designing software, Gene Fisher, FISCHER, Gene Fisher primer designing software. You can use Prime 3 Perfect designing software, Prime 3 Perfect primer designing software. You can use Prime 3 Out. All these are freely available. Prime 3 Out, etc. So there is one web page by the name 123 Genomics. When you type it on the Google, 123 Genomics. So a lot of series of softwares will come and most of those softwares are freely available online, right? One precaution you have to take when you are going to design primer for the expression analysis is that when you download the gene sequence, so most of the time that gene sequence will have untranslated regions also, and when you design primer using these softwares, so maybe your forward or reverse will go into the untranslated region. So in that case the expression which you are going to get is not the right or doesn't depict the expression of that specific gene. So what you have to do is first you have to find out where is the ORF of that particular gene and try that your primer should sit between the ORF of that particular gene. Is it clear? This is one common mistake I have seen in almost most of the cases which people they are using for the expression. So one primer will be in somewhere 3' UTR region, other will be in between the gene. So that is not the correct depiction of the expression of that particular gene. Right? So these are some of the softwares. When you feed your sequence, easily you can designate and you can design. One precaution you have to take, that the amplicon size should be between 200 to 300 base pair. Now you have to tell me that why amplicon size should be 200 to 300 base pair, why not 1 kb, 2 kb, 4 kb? The answer is, it's not that if you take 1 kb, so it will not show the expression of that particular gene. It will represent the expression. But then with the length, because your cyber die is limited, so cyber will go on intercalating. So maybe within few cycles, all your cyber will consume and then it will be unilateral PCR. Right? So in order to save, in order to get more accurate results, we prefer to have an amplicon size of 200 to 300 base pair. Right? So other than this chemistry and then oligos, next is some of the accessories which you require. And you should know because these accessories plays very important role when you plan for real-time PCR. First one is semi-skirted 96 well plate. So this 96 well plate comes in different forms, skirted, semi-skirted. So based on the instrument, some of the instrument they prefer to have semi-skirted, other they used to have skirted plate. So you need to have a 96 well plate which should fit into the platform which you are going to use. Then you should have a small thin plastic pyroclastic sheet, right? There is a small thin sheet. Sometimes we used to call it as aluminum sheets, sometimes we used to call it as pyroclast sheets. So this we are going to keep over your 96 well plate. The idea is that once the temperature goes 95 or 96 degrees Celsius, so your reaction will start evaporating. So that evaporation part should not come and lie on the surface of your detector, because once or twice if it comes and is stuck onto the surface of the detector, next time your detector will be damaged. And if it damaged, then cost I have already told you. So this is packed. And another thing is, since the reaction volume is not very high, so we don't want to have a different reaction volume per cycle, right? So it should be packed. So once you pack this, then you need to have a roller. There is a specific roller, so you can roll and then you can remove all the air bubbles which is present in between your thin sheet, so that no air should enter or no air should come out, right? And last, you need to have a 96 well plate spinner. So it's like a spin wheel. So you put your 96 well plate, just go for a spin. The speed should not be very high. 2500 to 3000 RPM is more than enough. Just run it so that you can accumulate all the reactions at the bottom. Now your plate is ready to be put inside the instrument. So these are some of the accessories or the requirements which you have to arrange before planning to go for real-time PCR, right? Now let's have a look on the workflow chart, how to execute the reaction. Workflow, right? I think before workflow, let me introduce you to different types of expression, because if you don't know the expression, there is no meaning to do real-time PCR. So can any one of you tell me how many types of expression are there? Expression of gene. Yes, any one of you? It's relative. So basically we have three different types of expression. The first type is called as absolute expression. Absolute expression means, how many of you have done Bradford method or Lorry method? So what we used to do is, you are going to have a known OD and then you are going to plot that unknown OD onto the standard and it will give you the concentration. So in case of absolute, we have a standard graph. You are going to have an unknown CT value. CT means threshold cycle value. That you are going to plot on the standard, and that standard will give you the number of transcript which is present in your reaction. So the expression which is calculated by using the standard graph is absolute expression. There is another expression which is called as relative fold expression. Relative fold expression means, here you are going to quantify the threshold cycle value of all the reactions, and then you are going to present your result in terms of or in comparison to the expression of your control, right? So for control sample, we are designating a number as 1. So if control sample has an expression of 1, how many times the expression in case of your treated sample? That is relative fold expression, means expression with relative to the control, right? So if control has 1, how many times expression increases or decreases in case of your treated? So if you calculate like this, this is called as relative fold expression, right? And third one is digital fold expression. Digital fold expression means we are doing transcriptome, right? And we are getting almost more than crores or lakhs of transcript in our data, right? Now, suppose there is a gene X and gene X has some, let us take 100 transcripts. It means this 100 transcripts out of total transcripts, if I calculate this, this is digital fold expression. Means how many transcript of a specific gene comes in a lot of total. That signifies the digital fold expression of that particular gene. Digital means by instrument read or the read of the instrument that we have analyzed, which has been created digitally, and then we have counted the number of transcripts, and in a mixture of transcripts, a specific gene has this much number of transcripts. That is the digital fold expression. So expression has been broadly classified into these three different types, right? Now, in the workflow, the first thing is that your tissue. So let us take two different tissues. So one is from your control sample, other is from treated. There is a precaution when you are going to collect tissue for the real-time expression analysis. The first thing is when you are going to collect the tissue, it should be properly washed. Once you give a cut, immediately it should be wrapped and it should kept inside liquid nitrogen or dry ice. Many times I have seen people immediately they will go to the field, they will cut, they will put on the ice, they will take it out and then they will use it for the RNA and then expression analysis. You will not get the exact result. That is wrong way of collecting the sample. So the right way is give a cut, before cut wash the tissue, take tissue something, you dry it, cut, immediately wrap it in some thin sheet, keep it inside the dry ice or liquid nitrogen. Alright. So once you have collected the tissue, so the next step is you have to use it for total RNA isolation, right? The method I have already told you. So when you run your total RNA which you have isolated by the desired kit onto the gel, we expect something result like this. There should be a prominent broad band, there should be a small band, and then there should be a smear. So this smear basically signifies your messenger RNA. These two prominent bands signifies your 20S and 18S RNA, and then there will be a plumbing here at the bottom. This will signify the non-coding RNA, right? Now, many times it happens that if you have not taken precaution, your RNA is partially degraded or degraded. So in case of partially degraded, result will be something like this. There is a prominent band, there is a small band, then there will be a smearing, and then here you will get very prominent band. It will be something like this. It means the RNA which you have isolated is partially degraded. If it is completely degraded, it will be something like this. It means the RNA which you have isolated is completely degraded, nothing is left over. And then you can validate this by using any of the technique or the instrument which I have listed. Is it okay? So once you have isolated RNA, next is Cdna synthesis. I have already told you the kit, and then I have also explained you that when you run your cDNA, we expect a light smearing with a prominent band. We expect a light smearing with a prominent band, that is best quality RNA, cDNA. And once you check that your cDNA is of good quality, immediately you have to quantify this. Now, there is two school of thoughts. One group of researchers, they prefer to quantify at RNA itself, and then once they quantify, they will take equal concentration of RNA. There is another group who loves to quantify at cDNA, and then they will take equal concentration of cDNA for the reaction. Now, you have to tell me why there is a need to take equal concentration of RNA or cDNA. Expression? Exactly. Because if you take more or less concentration of this, means you are biased. If you take more concentration of RNA, definitely number of transcripts and expression will vary. So at this level, or this level, you have to check, you have to unify. The recommended method is at cDNA level you have to quantify, because if you quantify at RNA level, we don't know what will be the efficiency of conversion of RNA into cDNA, right? So best is at this method you have to quantify. So once you have a cDNA with you, next is reaction mixture. So basically I'll tell you the protocol about the cyber, which is very commonly used. So in case of cyber, we are using, or this cyber comes in 2x form, right? So first, the volume of the reaction. So the most preferred volume of reaction in case of expression analysis is 20 microliter. If you are expert and you have large number of samples, you can decrease this volume to 10 microliter also. It will not have influence on the result, or you can increase up to 50 microliter also. But the most optimum volume is 20 microliter, and since this is in 2x cyber form, so here you have to take 10 microliter, right? Next is your primer. And when I write 2x cyber mix, it means enzyme and other things are already mixed with your cyber. Then primer forward. Basically this is 10 micromole concentration we used to receive. You have to take 0.2 microliter forward, and then you have to take 0.2 microliter reverse. Right? 0.2 microliter of forward primer, 0.2 microliter of reverse primer. Next comes your cDNA, which you have synthesized. It's recommended that one should take 20 nanogram per microliter cDNA per reaction. But then we have standardized anything between 20 to 100 nanogram that you can take, you will get good result. 20 to 100 nanogram per microliter. This is per reaction I am telling you. Then you have to make up the volume to 20 microliter. Let me repeat once. I told for reaction mixture, so 2x cyber you have to take 10 microliter. Forward primer, which you have designed and synthesized, this is basically in 10 micromole formulation, that you have to take 0.2 microliter. Reverse, again you have to take 0.2 microliter. Then cDNA, whatever you have synthesized, quantify it, make it into 20 nanogram or maybe 100 nanogram per microliter, and then take 1 microliter out of this. Okay, then make up the volume to 20 microliter. So this is the reaction mixture for, or this is for one reaction volume.
Making up with what, sir?
Water. Make up the volume with double distilled water or nucleus free water, right? So this is per reaction. Now how to calculate how many reactions I have to keep? This is itself a very tedious process, because I don't know how many reactions I have to keep. So for that calculation there is a chart, reaction chart. You have to make a table like this. This is called as reaction chart. Okay. So one side you have to write the treatment, other side you have to write the genes, right? So genes, let us take like G1, G2, E1 like this, and then treatment, like let us take 2, control T1, T2 like this you can make. Now in case of real-time PCR, the thumb rule is you have to keep reaction in 3 biological replicates minimum. There should be 3 biological replicates, and if your lab is rich one, you can take 3 technical replicates also. But at least minimum 3 biological replicates is recommended. So 3 means I can write here 3 like this. And then you total it. It means it is 27. So what I'll do is 27 plus 1 either I'll make, and if I am putting regularly, or if I am doing this regularly, this real-time PCR experiment, exact I will keep 27 reactions. Now if you recall here, I have written G1, GU, but in one case I have written E. What does it mean? Endogenous control gene means whatever CT value you are going to get, that you have to manipulate, you have to normalize using the endogenous gene, right? So endogenous gene is basically a gene which doesn't show any change in the expression with respect to the environmental factors. So that is endogenous gene. For example, in case of crop, some of the preferred gene are like you can use actin, beta actin, elongation factor you can use GAPDH, you can use globulin. So these are some of the genes which are very popularly used as an endogenous gene for real-time expression analysis. Right? So once you calculate the reaction, you know the volume, how many or what is the volume of ingredients which you have to take per reaction. So next is how to make a reaction mixture, because this is very dicey. If I have to pipette these all ingredients one by one, I am a human being, so maybe one or the other part I will commit mistake. But there is a specific method through which if you use that method, the chance of error will be very less. So let's see how to pipette it out. So in case of pipetting, the first thing is you have to take a 1.5 ml Eppendorf, right, and add this cyber. Now here if you see, this cyber is common among all the reactions, right? Cyber and the water which you have used. So what I will do is, first I will add the cyber and the water into my Eppendorf tube. Okay. Then I have two treatment, that is control and then T, right? What I will do is I will divide this into two equal parts. Then I will add the respective cDNA, cDNA of control, cDNA of treatment. Again I will mix, and now I will divide this into the number of genes which I have taken. Now I will divide this into the number of genes. Suppose if I have taken 2 genes, so this volume will be divided into 2 equal parts. If I have taken 3 genes, 3 equal parts. Right? Now once you divide this and when you quantify, your volume should be exactly, it should be exactly 60 microliter. Right? Why 60 microliter? 20 into 3, and 20 is your volume per reaction. So 3 replicates. If it is exact 60 means your pipetting is perfect. Now you are ready with loading the sample onto the semi-skirted plate. Right? So once you have done pipetting like this, Eppendorf is ready with you, volume is 60 microliter, then take the skirted plate. I will show you the plate when we will go to the lab. So plate will be something like this, skirted plate. What you have to do is, in a parallel way you have to load your sample. Like suppose G1, this should be my 20 microliter, 20 microliter, 20 microliter, like this. Then G2, E1, like this you have to load. Is it okay? So the check is 20, 20, 20. It should not be 55. It should be exact 20. Because if this volume doesn't match with the 20 microliter, it means the concentration doesn't match with what we have written here. It means the reaction is not appropriate. It means when you feed it into the instrument, you will get abrupt CT values. Is it clear? So this is self-check. If you get exact 60, fine, it means everything is fine. So once you load this in all this, then you have to take that parafilm sheet and then you just put parafilm sheet from one side, from left to right. just stuck one side and then slowly keep on pasting over this. So once you paste the parafilm, then I have written roller. Take a roller and then roll it. If you don't have a roller, you can prefer to have a glass rod or the pencil, anything you can use. With full power, roll it so that all the air bubbles which are present here, all these bubbles should come out. Once your plate is ready, then I told you have to feed it into the plate spinner. You keep it, run at 2500 or 3000 RPM so that all the reaction mixture should assemble at the bottom. Now your plate is ready for feeding inside the instrument, right? So once you feed inside the instrument, the rest part instrument will do. But then the reaction cycle that we have to feed from our side. Let's have a look on the reaction cycle. Basically, two different types of reaction cycle are preferred in case of quantitative real-time PCR. One is called as two-step cycle. One is called as two-step cycle. Other is called as three-step cycle. Right? The difference is, in case of two-step cycle, the first step is that you are going to have a denaturation that is 95 degrees Celsius for 4 minutes. Some lab they prefer to use 3 to 4 minutes. Right? Then you can have a 94 degrees Celsius for 45 seconds. So 30 to 45 seconds. Then you should have a TM for 20 seconds. The range is 20 to 30 seconds. And after that, there is a plate read. There is a plate read. Plate read means your detector will come, it will sit on your skirted plate, it will take the reading of the CT values and then it will go out. Right? And after that, this will be cycle 39 times. So total there will be 40 cycles. Right? So this is called as two-step cycle. There is a three-step cycle where all parameters will be same. The only thing is before plate read, we are adding an extra step which is called as 72 degrees Celsius for 5 to 10 seconds. And after that plate read will come. 72 degrees Celsius for 5 to 10 seconds and after that plate read will come. So this is called as three-step cycle. Rest other features will be same, parameters will be same. The advantage is, few people they claim that when you give 72 degrees Celsius for 5 seconds, so there will be a stabilization and then when detector comes, so that will give the exact rate of fluorescence. But in this case, since simultaneously cycle is going on, so maybe that will be not so exact. But it's not case like that. In both the cases, we have experienced same result. Right? So now this is a cycle which you have to feed into the instrument that I want this two-step cycle or three-step cycle. So once you feed this, then instrument will ask you which one is your control, which one is your treatment. That you have to feed. That well number 1 is my control sample, 4 is treatment, and when you feed all these informations, then your most of the work is done. Just run it and then instrument will give you the result in terms of CT value. And when you see the graph, it will be something like this. So different reactions will show different type of graphs. But if you get a sigmoidal graph like this, it means most of the primer worked nicely and you got the perfect CT value. So this particular portion represent your CT value. Right? So now if once we got this CT value, then there is a specific mathematical method to calculate this. even though most of the instrument they have inbuilt software. So this software will calculate and they will tell you what is the expression of your gene. But then manually also you should know how to calculate the relative fold expression, right? So let's have a look, even though it will be difficult to explain here because this is Excel based function. But then I will try, and when you go back to your hostel, easily you can feed and then you will get that result. So this is last part, and then I'll stop after that. We'll go to the lab and we'll do the practicals, right? So how to calculate the relative fold expression based on the CT values. Calculation of relative fold expression. So the method we used to follow is called as Pafal method. Pafal 2002. So he was the gentleman who first discovered the mathematical equation for the calculation of relative fold expression using the CT value. All right. So now let us take, you have a gene 1 control, gene 1 treated, endogenous gene control, endogenous gene treated, right? Now these are the replicates R1, R2, R3. So the first thing which you have to calculate is the average value of this CT. CT average, or we also used to call it as delta CT. Okay, so first you have to calculate the average. So average, I think almost all of us know how to calculate the average. So once you calculate the average, then there is a term which is called as delta CT. Delta CT means differential of threshold cycle value. So what we are going to do is, this value like G1C, suppose if this is A, this is B, this is C and D. So this value comes when you subtract this control of gene with the endogenous control, right? Means A minus C is your DCT value or differential CT value, okay? Differential CT value or DCT value, whatever you want, you can pronounce. It means the CT value of the gene minus the CT value of same treatment of the endogenous control gene. This will be B minus D, and these two you are not going to calculate because that endogenous gene was just used for the normalization. So once you calculate the DCT value, then you have to calculate differential of CT value, which is called as DDCT value. So for the calculation of DDCT value in Excel, there is a string of pound, right? What you used to do is, suppose the address of this particular cell is, let us take 16, right? So it is A16. So here you have to feed the formula this value. Let us take this value is something Z. So Z minus, then you have to use a string. Then you have to feed same this A16. It means Z, the value of this minus itself. So what will be the value of this? And the string means once you drag this string will make all these value constant, right? So the address of this minus string address of this, so it will be 0. Now you have to drag this. Okay, so this value is called as DDCT value, differential of CT values. And last one is 2 to the power of minus DDCT value. 2 to the power of minus DDCT value. So power that you know in Excel easily you can put. So 2 to the power of minus DDCT value means this one. This will be equal to 1. And when you drag this, this will come some values. It will be either more than 1 or it will be less than 1. Okay. Now our control has a value of 1. Treated has a value of something which is either more than 1 or less than 1. Now if it is more than 1, it is upregulation of the gene. If it is less than 1, it is downregulation of the gene. And this 2 to the power of minus Ddct, this is called as relative fold expression. So in your manuscript, when you are writing something, you have a liberty to use 2 to the power of minus Ddct. This also you can write, or you can write relative fold expression. Relative fold expression, and then in bracket you can write 2 to the power of. Either way you can represent your result. It means in comparison to 1, what was the fate of treatment on the expression of gene which you are going to target. So when you feed this formula into the Excel and when you drag it in one go, it will give you the result. So this was the method discovered by Pafal. So we also used to call it as Pafal method, right? So I think even though time was limited, so I was very short. Few of the things I skipped purposefully, but then I think the gist of this experiment, that where to use, how to use, and how to calculate the result. So that I think I conveyed. The expression when you are writing something, so the best sentence to express the expression of any gene is like the expression of gene X was upregulated, whatever times you have to write, that you can write in bracket, maybe 3 times upregulated as compared to control. This is the best sentence people they prefer to write in the manuscript. So in one sentence, you can explain the fate of expression of your gene. So I think I will close here and then we'll go downstairs, and whatever I have explained here or I have tried to explain, let us do practically with your own hand. Thank you.
So here we have taken one gene and one endogenous. So gene is our MAP kinase, and endogenous gene which we are using here is beta-actin, and we are going to do it in wheat sample. So here the treatment is heat stress. We are giving heat stress to the plants at vegetative stage and at different stages. So we are isolating the RNA from leaf tissue, and then we are going to keep the seed. So then cDNA is already prepared here. So we will keep the reaction. So here total the number of reactions which we are going to keep, it is in duplicates, it is total 8 reaction. One control, one treatment, then one gene and one endogenous gene. So and in duplicate, so total 8 reactions we will keep. So first we will take water, just in this reaction 1.5 microliter, if this is epandroactive. So here we will make a first reaction mix we will make. Okay, so please come. We will add 80 microliter of water. Here we are using nuclease free water and a cyber green. Okay, so 80 microliter of cyber green and nuclease free water, it is 65.6 microliter which we will add. So this is nuclease free water. Take 60. First set your pipette. How much it is? 80 cyber. This is cyber. So since this cyber, it is light sensitive thing, so always the tube which the company people provide cyber, it is in brown tube, this dark color. If we are transferring it in some transparent tube, so it should be covered, otherwise reaction will usually lights please switch off the light. So take 80 microliter of this cyber in this tube. You know how to pipette it? Be careful, your tips should not touch anywhere else. Always try to put it at the bottom of your tube. Okay? discard this two. Now you have to add water, nucleus free water. So 65.6. So first set your pipette at 65.6. 65.6. Okay, so you add it. So here we have make a common mix of cyber and water, because these two things are common in your all the samples. Okay, whether it is endogenous gene, whether it is heat stress, or it is control, or your gene. So for all these things, these two things are common. When you come to later, so in some case your primer will be different. Like for actin, you have to use actin primer. For MAP kinase, you have to use MAP kinase primer. Okay. Then for the sample or cDNA which you are taking, so for your control, you have to take cDNA which is prepared from the control sample. And for treatment, so you have to take cDNA which is prepared from the treated sample. So that thing will be variable. Okay, so first we will make this. Have you add water? So first add water. So only the cyber and the water, these two things are common in both the means all the reactions. So we make one common reaction of all these things. When we are working on RNA or RNA related work, so we have to be very careful that, because RNA is very sensitive thing. cDNA, it is stable as compared to RNA, but still we have to take care of all these things. So always wear gloves, and your place should not be contaminated with anything. Okay. So have you mixed it? So for mixing, you can spin it. Spin it so that your water and sugar will be mixed properly.
Because it's balanced now.
Not balanced. Need one to.
Let him go.
Okay.
Now only these two things are common in all the reaction. Okay. Now you have 4 different kind of reactions. Okay. So we will divide this mix into 4 different tubes in equal quantity. Okay. And then we will add, in some case we will add wherever required, the primer and the cDNA according to our samples. Okay. So in each tube you will divide it into 36.4 in all these four tubes. 36.4. Set your pipette at 36.4. So whenever you are going to do this real-time expression profiling, before going to perform directly, means set your experiment, first you have to calculate it and write it in your front, so that it will be easy for you to set the reaction. Okay. So just distribute this in all the 4 tubes, 36.4 in each tube. Try to keep it at bottom, and don't pipette it with so many times. Then it will create bubbles, and sometimes it happens that it may be stuck in the tip, so loss will happen. Okay, we have to be very careful. Try to keep it at bottom. Now see, we have forward primer, reverse primer of 2 different kind of gene. Okay, so one sample from control and one sample from the treated have the actin gene primer, forward and reverse, and another have MAP kinase. Okay, so here we keep like this only. This is your actin control, and this is your actin heat stress. Okay, so in this case, in in these two tubes we will add actin primer, and in these tube we will add MAP kinase primer. This is heat stress, this is control. Okay, so you have to add 1.6 microliter of forward primer. This is forward primer, actin gel. So add 1.6 microliter of this actin in both the tubes. So
0.8 will be there, no? 0.8 of forward, 0.8
okay,
of the total.
so 0.8. Just which pipette you are using? No, so 0.8 microliter of forward primer in both the tubes. Be careful while 0.8. Be careful if your tip, you may touch it, but change in the next case you can change it. Okay, otherwise the primer will be contaminated. Try to drop it at the bottom. Not change this tip, because because this tip is already touched here. Okay, if same tip we will use for this, so this stock will be contaminated, because you have so many other things are there, water cyber is there, so it will be contaminated. So better change. Same primer, forward primer, acting. This one the heat stress, this is control. Now reverse primer of acting. And same, this is the reverse primer, 0.8 microliter, same. Okay. Because the amount is very less, so whenever you take it, always check your tip whether it is coming or not. Sometimes happens that it is not coming in the tip, so always take care that it is there or not. Okay, so up to primer, this our actin gene is okay. Okay, now MAP kinase forward primer. This is the forward primer. Okay, so up to primer, now it is okay. Now we have to add Cdna. Last step, template addition. That's all things are added. Now this is the control, this is the addition. There is the control Cdna. Cdna, how much you have to add? 2 microliter in each. Okay, so 2 microliter of Cdna we will add into. This is heat stress. तुम क्या ले रहे हो? 2 microliter of Cdna will add. When you, sometimes happen that while doing this, if your tip touch anywhere else, then immediately change it. Otherwise, it may contaminate your stock solution. So that will be more expensive, because okay. So Cdna for control is over. Now Cdna for heat stress. Now all things are added. Now we have to require that it's all the components should mix properly. So we can spin it. Now this is what we call about the semi-skirted plate, 96 well plate. So always nothing except this type of plate. Okay. Now here reaction is prepared. Now we have to distribute it in the pairs. Okay. So before going to distribute it, we have to draw it on our copy that in which well what we are going to add for our record. Otherwise, sometimes if suppose we forget in which well what we have added, so we may confuse because we required calculation of each well. Okay, so it's better we draw it in our copy that in which well what we are going to add. So for that, what we can do, we can make similar there is 12 column 8 rows. Okay, the same plate we have made here. Okay, 12 columns 8 rows. Here it is written 1 2 3 4. Here A B C D E F G H. Okay, A B C 1 2 3 4 like this. Now suppose we are going to add here control gene. Okay, control gene, then heat stress gene, heat stress gene, then control actin, heat stress actin, heat stress actin. Okay, so here now suppose if I forget, still we have a record, we can trace our data. Okay, now we have to distribute our sample in the same way. Okay, so 20 microliter. So 20 microliter in each tip. There is a well. You can see there is a well. Okay, so we will put 20 microliter according to our this thing. Okay, so we will distribute our sample in 20 microliter. First is control gene, control map. This is control gene. 20 microliter in A1 B1. This one. This is total 40 microliter. So 20 microliter in A1, 20 microliter in B1. Try to put it at the bottom. Same tip you can use here because there is only sample distribution, same thing is there. So for one sample, one duplicate. A1, B1. So this is our control gene. Okay. Now heat stress gene. It is A2, B2. While putting our sample into the plate, we have to be very careful since here only 8 samples are there. Sometimes we are using 96 well plate, 96 samples. So we have to be very careful, should not miss a single well. Otherwise, whole experiment you will be confused whether what we have loaded, and so you will not get the proper result. So we have to be very careful. It required means you have to be very focused while loading your sample. Now actin control. A3, C, B3. And same information we have to feed in the machine also. The real-time machine also same information in which well what we are going to feed. So that we have to feed there also. Heat stress actin, endogenous gene. A4, B4. Now your reaction is ready. Now the next step is we have to cover it. Otherwise, it may evaporate when you keep it because temperature, the initial denaturation temperature is 94, 95.
95.
So, up to that much high temperature, there is possibility that it may evaporate and it may destruct your machine. So, it's better. So, it is a transparent, and always we prefer to cover the complete plate. Okay, so this is the transparent sheet. So we have to cover it like this. So we have to take care of that. There should not be any air bubble. Okay, so we have to keep it like in such a way that there should not be any air bubble. We press it properly. Seal it. Now this extra part we may cut it like so. Another idea is also that why we should cover it this complete plate, because the this time we have used only 8 wells. Rest wells are fresh. So if it is covered, so we can use this plate further also. This rest of the well which is not used, so that wells we can use because it is not contaminated. Okay. So this plate is now ready. Now we can spin it. There is 96 well spinner. Why spin it? So that while putting, there is possibility that some sample may be at the wall. So all will be come at the bottom. Now your reaction is ready. Now we will put this plate in the machine, set the program and run, just like PCR. Now I can see all the samples are at bottom. There is no air bubble. Yes, it is not. Sometimes happens that some samples may be stick at the wall or maybe air bubble there. So by spinning it will okay. Now we will keep it in the machine. Please come to the next room. before keeping our plate in the machine, first we have to set the means whatever our experimental planning is there, we have to feed it. So I've opened here means already the protocol feed here, not making because the steps whatever required 95 for 3 minutes, then 95 for 10 seconds, 60 for 30 seconds, 72 for 10 seconds, then plate read, then go to 39 means step 2 for 39 cycle modes, then melting curve 65 degree, then plate read, and then the programming. So this is already fit, so I am not going to feed all these things. Okay, now we have to give the information of this plate. In here are all unknown. Okay, we know that. Now the next is the next step is open because we know the program is set. We have dictated these will set the samples. Now open them. So see, in plate there is a cut mark here. Okay, we have to keep the plate in this way. Okay, now close the lid. Now lid close, and here start run icon come. Okay, so we will